Hello, everyone. My name is Danielle Tallman, and my presentation will be covering the modulation of cytokine patterns and microbiome during pregnancy in IBD. I'm excited to present this article since it covers a growing health concern in the United States. There are many publications covering irritable bowel disease, but most focus on the individual's health with the disease pathogenesis and mechanisms of the disease. This article stood out to me since it involved the mother's health outcomes with pregnancy. In this presentation, the research of how pregnancy affects women diagnosed with IBD will be discussed. The main focal point is how pregnancy affects the future health of diagnosed women with two common types of IBD. To give some background understanding of the topic, let's go over what IBD is. IBD is an umbrella term used to cover diseases that are caused by inflammation of the colon and/or the gastrointestinal tract. Even though there are several, this study has participants diagnosed with Crohn's disease, abbreviated as CD, and ulcerative colitis, abbreviated as UC. Crohn's disease can have various areas of inflammation throughout the entire GI tract. Ulcerative colitis is different than CD since it primarily affects just the colon rather than other gastrointestinal areas. Another major difference is that CD can affect multiple layers of the intestinal lining, while UC only affects the innermost lining of the colon. I have noted that 31 patients in this study have been diagnosed with CD, while 15 were diagnosed with UC. This will be covered more later in the presentation. I was curious on the data that we have on the growing population diagnosed with IBD. According to the CDC, 1.8 million Americans were diagnosed with IBD in 1999. In 2015, 3.1 million were diagnosed. The CDC noted that there was a higher prevalence of people diagnosed if they were over the age of 45, Hispanic, non-Hispanic white, adults with less than a high school education, unemployed individuals, and individuals living in poverty or suburban areas. There are slightly more women diagnosed than men. On this chart, I have highlighted the estimated number of women as well as the ages women could be pregnant. Over 500,000 women would fall into the category of being of a birthing age and diagnosed with IBD. Many of these women may want to start a family, but they may hesitate since the health of themselves has a high influence of the health outcomes of that pregnancy. That is why IBD research, like the study done in this presentation, and many other studies are relevant areas of research. To expand on the research from the previous slide, I got a fantastic supplementary article recommended to me by one of my peers showing the importance of this research. It not only goes into detail about the genetic factors already known about IBD, but goes into further detail about how pediatric populations are now being diagnosed as well. I have this article listed below and highlighted in the citation section if you are interested. And in this is just an additional image from one of my supporting articles that emphasizes the importance of a mother's microbiome on fetal health. In this article, it starts by covering normal in a regular pregnancy. A mother goes through a lot of physiological changes while the fetus develops. The modulation of the mother's immune function allows for the fetal major histocompatibility complex to be developed and to prevent the mother's usual immune defenses from attacking the embryo and fetus. It also allows for immune tolerance to flux to support the different stages of pregnancy. For example, for successful conception to occur, the Th1 inflammatory cytokines must be present at the maternal-fetal interface. A shift from the Th1 to the Th2 occurs during the majority of pregnancy since it has the most tolerance. Just before birth, it reverts back to Th1. This is a natural process that shows that it is necessary for a mother's immune system to be altered during pregnancy. This study is not only observing differences, it also tries to make as many comparisons to truly distinguish what is quote-unquote normal for a pregnancy. Which leads me to the methods used. The researchers used two groups to compare for this study. The control group were patients not diagnosed with IBD or any other gut inflammation. The test group consisted of the individuals diagnosed with either UC or CD. For testing, fecal samples and peripheral blood draws were collected from each participant. 46 patients were a part of the IBD group, while 179 healthy controls were participating. The times of collection were pre-pregnancy, the first, second, and third trimesters, as well as postpartum. For cytokine testing, blood was collected, spun to the serum level, then tested with an ELISA assay kit. It's the Ready, Set, Go by eBioscience kits. The kit tested for 12 different inflammatory markers. As for the microbiome testing, fecal samples had DNA extractions performed using the PowerSoil DNA isolation kit by MoBio. Then the V4 region of the bacterial 16S rRNA was amplified by the extracted DNA. They used 515S and 806 bar-coded primers following the Earth Microbiome Project protocol. After additional purification, it was then sequenced using the Illumina MiSeq platform. Analysis was performed using QIIME-2. The overall results of the study shows the pro-inflammatory cytokine levels decreased during pregnancy for IBD patients. The researchers compared pre-pregnancy levels to active pregnancy. It was also observed that the IL-10 and IL-6 cytokine levels increased with healthy pregnancies, but did not increase in patients with IBD. This shows that pregnancy in healthy women are associated with specific changes in cytokine levels that are not present in women with IBD. The article used some recurring terms that I wanted to briefly discuss before we look at some figures. T1, T2, and T3 is referring to each trimester of pregnancy. Also, they use some scales that I was not personally familiar with before reading the study. They use a weighted and unweighted beta diversity scale, which measures the difference between two microbial communities. They also use Faith's phylogenetic differences measures for the alpha diversity and Pylew's evenness measure, which measures how evenly species are distributed in alpha diversity. The overview of this figure is that it shows patients with UC and CD do have their own cytokine values that are unique to each. The green represents the UC patients while the red represents the CD. This is just the data from each active trimester of pregnancy, so it does not include the pre-pregnancy or the post-pregnancy. And it does show major differences in the inflammation. Some notable comparisons between trimesters were for cytokines IL-12 and IL-21. There was more cytokine values for UC than CD. But it is interesting to note the decrease from trimester 2 to trimester 3 for UC. On the contrary, you can see the values of IL-21 jump considerably for the UC from trimester 2 to trimester 3. For comparing differences of UC to CD, the values of IL-9 stay consistent in the CD patients, but slowly rise in the UC patients. Then for the IFN gamma, CD stays consistent once again, but the levels decrease with UC patients for each trimester. I definitely wanted to highlight all of these significant values because per my own personal preference, when there is a lot of information on one slide, I like to personally hone into just what is important for this study. And then you guys can always look back and see the comparative values as well. Now we can see the comparison of the IBD patients and the control group, both during active pregnancy. Here we see that the IL-6 and IL-10 levels mentioned earlier do not rise in IBD patients, but they do in the healthy pregnancies. IL-10 and IL-6 both have similar ranges through each trimester. TNF-alpha shows very comparable values of the control and the IBD trimester values. Then, seen for the differences of each test group, IL-5 shows a lower concentration for IBD than the controls. IL-8 is greatly increased for IBD versus the controls. And the same for the IFN gamma values. It can be seen on the visual. I really appreciate that it shows how there can be differences in the trimesters and between the IBD and healthy patients. It does show some inflammation still occurs, which we would of course see because of inflammatory bowel disease being an inflammation issue. But it does show a lot of comparable or acceptable ranges that is viewed in an average pregnancy compared to this already inflammatory disease. For the comparison of microbial diversity, we already know from previous analyses that the microbiome of an IBD individual is greatly different from a healthy person. With this diagram, we are getting confirmation that pregnancy does not change the microbiome of an IBD patient. This correlates with the inflammation patterns seen in the previous slides, since the microbiota are essentially still existing in an inflamed environment while pregnancy occurs. Figures A and B are measuring the beta diversity changes for each of the 5 times stool samples were collected. So the pre-pregnancy, trimester 1, trimester 2, trimester 3, and the postpartum sections as well. And if you notice, there are color-coded for A, B, C, and D. Red being pre, T1 is blue, T2 is green, T3 is purple, and postpartum is orange. So the same is also shown for the alpha diversity in figures C and D. If you look closely, there are some black arrows on the scatter plot. Those patients were taking a course of antibiotics during pregnancy, so their results are still included since they began the study, but it's noted that they are kind of separated because of the non-expected antibiotics that they went on during their pregnancy. Figure E, which is on actually the next slide. Oh, no, it's not. Well, the recording won't let me go back, but basically, figure E just simply showed some of the main phylum present in the samples and how they remain about the same during pregnancy. So now we are on, let's see, slide 17. The study noted that the microbiome from pregnant patients with IBD is less diverse than in the healthy controls. IBD patients share more in common with other IBD patient microbiomes than the healthy controls, as seen in figures A and B on the screen. In figures C and D, they had lower alpha diversity differences per trimester and less evenness, which we will kind of get more into that later on. Okay, this is the last segment of the previous figure that shows the richness of the microbiota recovered and then how it reduced over time for the IBD patients versus the controls, which you can see with how wide it is on this whisker plot that's happening right here. You can see how the red goes from being a big, thicker chunk of red, and then it slowly becomes less. It starts to get a little bit better during the third trimester, but the control shows a little bit more evenness and consistency throughout. So now we're going to begin a bit of the discussion. There was a lot to look at through this entire study, and honestly, there was a lot of figures that, even though they were really cool and important to some particular parts of the study, I did purposely exclude and just focus more on the comparative aspects of this study done. Those figures are in the original paper, and they more look at the different species and differences of the fecal collections, versus my particular focus of this presentation was to look at the inflammation compared to microbiome changes. So if you are interested to see more of those specific images, I would recommend going back into the article, and I'd be interested to hear what your thoughts are with those different visuals that I didn't show in this presentation. So for the researchers, for what I did show, the researchers noted that it is normal for cytokines to raise in any pregnancy, as noted before. I just want to really reiterate that. But here there are the common cytokine values that always increased was IL-6. So we've seen IL-6 increase in both the normal and the IBD. And that is just a very common marker that was seen just throughout this entire study. Since there are specific cytokines associated with Th1 or Th2, the researchers were really hoping to have a specific group, either the Th1 or the Th2 cytokines, to correlate with more of the inflammation during pregnancy for the IBD group or the control. But the data that they collected, well, there wasn't enough supporting evidence to solidify any particular claims. They did not know or have enough information to state which cytokines were reducing inflammation or which cytokines were reduced in the pregnancy. But once again, they only had enough data to highlight that IL-6 was the consistent marker that could be shown and have enough evidence behind. Noting the differences of the microbiome of each IBD subgroup showed that even though there are two different diseases, they become more common in pregnancy. Kind of as we noted before, they do have different levels of cytokine markers that are inflamed because they are two separate types of inflammation in different parts of the body. But it was just really interesting to see that as the pregnancy continued, they became more similar. So I quite thought that was an interesting aspect about all of this. And then overall, the researchers could see an increase in the beta diversity and a decrease in the alpha diversity of both the IBD patients and the control groups, which is kind of cool because it suggests that the modulation of the cytokines connect with the species recovered from the samples. But as I kind of mentioned before, I really wish there was some more evidence, but there's not sufficient evidence to state anything further besides that they just kind of noted these changes occurring with the increase of beta diversity and the decrease of alpha diversity. The alpha diversity was even more interesting to me to view, since the values were different in the T1, the trimester 1, but progressively became more similar later in pregnancy. So you can actually see on those graphs in T2 and T3 how they kind of equalize to one another. This helps to show overall that pregnancy does not disrupt IBD patients' microbiome negatively. And that can be something that is super exciting news for someone who is diagnosed with IBD and is just kind of concerned about their health and how that pregnancy is going to go through with them through each trimester of pregnancy. There might be improvements, so that's really exciting. In conclusion, there are normal cytokine changes that are normal in pregnancy. So that's just kind of restate what the studies found. The research has supporting evidence that IBD does not negatively affect pregnancy, and with certain cytokine markers, it actually improves during pregnancy. More research is needed to explore the shifts of specific species making up the fecal microbiome. Finally, more research is needed to correlate specific cytokines with microbial shifts. That's something that, as I was reading this paper, I really wished they had gotten further into this topic of comparing what particular microbes were actually changing and which particular microbes were maybe more associated with cytokine markers, but I feel like they need to do some more research with specific microbes with specific cytokines to get a more clear answer to this. Like I mentioned, I just really believe that this study was super beneficial for people who are suffering from IBD, and I would, as a person before reading this article anyway, I would have thought that, okay, if I have an inflammatory disease and I know that pregnancy causes inflammation, the last thing I would want is to have even more inflammation, also have even more immune dysregulation than I currently would have if I had that disease. So from that perspective, this is a really exciting study that shows really positive answers, because the fact that even though we didn't get specifics on anything, the overall picture that nothing could get worse if you get pregnant is overall a very positive message. So I really actually quite liked the results that were seen in this study overall. So I took a little time to think about some questions that not only was I thinking about, but maybe could be expanded upon as well. So the first question I came up with was, what was one finding that you found interesting? Because personally, I chose this article. I found the topic very interesting. I also, during some discussions, found that other people found this topic interesting. So were your questions answered the topic that you found interesting, or was there something new that you took from this article that you also find interesting? Second question, with the data in this study, what do you wish they went into more detail about? I already stated some of my opinion about I wish they had gone into more detail about what shifts of microbiome was occurring at different times of pregnancy. There, of course, are visuals of this, but essentially I really wanted to see more of a comparative study of the control to the IBD, because we basically got the information from this that IBD doesn't really change the status of where the microbiome is during pregnancy for people diagnosed with IBD, but I just felt like they could have gone a little bit more into detail about this. And then also, what is it like for the mother more postpartum? Like, how about, they have the postpartum fecal sample, how about a month out? How about 2 months after birth? So those are different examples I kind of thought about when I made this question. The third question is the amount of data collected in the study sufficient? I'm mainly pointing out that you can clearly see there are less IBD patients in this study than non-IBD patients. So I feel like that may or may not have changed some of the results of this study, but I am curious to see what you all have to say about that. The fourth question. should they have added other inflammatory intestinal diseases such as leaky gut or IBS? Like I said, they only included UC and CD in this study. So should they have just kept with one? Should they have looked at more to gather more people to potentially be a part of this study? Or would those be too many different confounding variables? I want to hear about that. The fifth and final question that I had made, which is kind of a long one, this study did not consider if it was the mother's first time being pregnant. So the participants could have been previously pregnant, or it could have been their first time. Do you think a first-time pregnancy results would have been different from a mother who previously had children? So if you have, the scenario of this woman who is diagnosed with IBD, she's on her third child. Let's say she's a part of the study, but then you have a woman who's first-time mother about, being pregnant, a part of the study. Do you feel that there may or may not have been any difference of inflammation, cytokine markers, different areas of this topic for each of those mothers? Or should they have kept things more consistent and looked at first-time mothers, or only continued second or third or fourth pregnancies for these women diagnosed with IBD? I had a great time talking about this topic. I thank you all very much for listening. I am super excited to look forward to the discussions that occur after this and to get some of your feedback on this. I love getting more comments about how I can improve as a presenter, so I am completely open to suggestion. Thank you all very much. Here's my citations. And there I have that highlighted article that I mentioned way earlier in this presentation. So thank you all very much, and have a great time responding to these very long and eccentric questions that I created.